attending the presentation today. I'm so happy today I can pass all the way until now with different tests. So here the first times I want to present my gratitude to all the committee members, for Professor Chen from National Taiwan University and our dean, Professor Leo, and also my advisor, Professor Chen Ulasan. Here I will present my thesis entitled Isolation and Characterization of Simple Sequence-Repeat DNA marker from marine herbivorous species. For these two species is milkfish and rabbitfish. And before my work is in freshwater aquaculture center in Indonesia, I work in ponds and hatchery of shrimp and some milkfish hatchery. And before you need a strong body to work in the field, and some are slimy and dirty also, and but change very dramatic to work in uncontaminated area, very convenient and clean, and this is maybe make me more brighter today than before. So I will say very lucky I can learn molecular biology in National Taiwan University. So we divide our presentation today because first is introduction, objective of study, experimental procedures, results, discussion, and then to conclude this presentation. So we hear my message is growth of fish meal and fish oil is stable and even decreased from 1990 until now, but aquaculture increase, and you can see here that capture fisheries is also stable. So why this can happen? Because these graphs answer these questions, because most of them is produced by freshwater fishes and also mollusks. So most of them is herbivorous species. Here 71% is dominated by carps or freshwater fish, and in 2008 catfish production is increased dramatically, especially in Vietnam. So because of this, because here is still in freshwater area, so we need to support for marine area. So we need to understand first the biology and aquaculture of milkfish and rabbitfish. These two marine herbivorous species as candidates. The first milkfish is, we know this is a species in a genus Canos, and this low trophic level eat plants, and the special ones is have many bone in its muscle, intermuscular bone. And then this fish is well-known farm fish in Asia, and the problem now is this fish is still underdeveloped for breeding program. So we here see that the production of milkfish is increased sharply, especially from 2006 and up to 2011. See, these only 3 countries with major production of milkfish: Indonesia, Philippines, and Taiwan. Maybe because in Taiwan the area is limited, so they are not increasing anymore for this species. But Philippines and Indonesia, because they still have a wide area for brackish water, they still increase every year. So also this table, these graphs show that even the productions increase, feeds used for this milkfish is still in the minutes of the production. It means they use lower feeds compared to other kind of species. So we can conclude here that this fish is efficient for use fish feed, And also this fish, because this marine herbivorous species, some of the cultural practice, they don't need to give them with feed, but sometimes they only need to fertilize their ponds. So this fish is in nature, they are always run in schooling, and then they grow in the estuaries, and after that they go again to the marine water. And because of that, they spread in many countries, especially in tropical areas. You can see here in Indonesia, Southeast Asia, and also until Japan. Also, the milkfish hatchery is quite cheaper compared to like carnivorous species like croppers. We just provide rotifers, phytoplanktons, and this is tapioca meals. Tapioca meal is very cheap. If you give the cropper the tapioca, your croppers will die actually. But for milkfish, because it's very hard, so they can, you just provide tapioca meals is easy. So the fingerling of milkfish is quite cheap. And also this picture provide the polyculture of milkfish and seaweed, and sometimes with shrimp and crabs, and quite profitable, even though this is traditional. But once is the problems of milkfish is have many ponds, so sometimes we need to remove the ponds. But after we remove the ponds, the price will increase, become twice. This show the ponding milkfish in Philippines. The second kind is Siganus canaliculatus. This is in this Siganus they have 22 species, and this fish is so like inhabit in shallow marine water and estuaries, sometimes in coral reef, and herbivorous, consumes macroalgae, most of them in food test, and that's special for Indonesia and Southeast Asian country. This fish is we use this for symbolize lucky for Chinese overseas. Chinese overseas in Southeast Asia, because their color brighter and their food test. So people overseas Chinese always use this as a lucky fish. Also in Imlek time. Imlek time is in the middle of the first months in the new year. And then also this also is successfully propagated fish. And the problem now is there's no breeding program of this fish. But recently the production of this fish, Siganus, is quite low. It's only 200 tons for the totals in 2003, and only the maximum is around 250 in 2007, 2008. Compared to capture rabbitfish around 62,000 in 2011. So this production is very low, only less than 1% of all the production in the world. So most of this fish, Siganus canaliculatus, is inhabit seaweed-rich area in the coastal area. Most of them eat this seaweed. And compared to milkfish, because these fish, they are not migratory or farm migratory fish, so we can see here that only a small place in Southeast Asia and near the Africa, but not in Latin America. And also the culture is quite simple, but this use more higher price live feeds like Artemia and also fish meal compared to milkfish. But the production is quite good. For example, from around 13 grams, it's become near to 100 grams or 181 grams in only 3 months. So gross fish production is quite good. They just provide copra meal, tapioca leaves, and seaweed and any others, but most of them use plants. So we can conclude here that seed for marine herbivorous species this can reproduce in captivity, also already reproduced in captivity, and they just feed in plant based fish feed, and disease is very less prevalence. You never read about the virus of this milkfish or rabbitfish, and also they can adapt in poor water quality. But now we encounter the problems. They have less genetic improvement in both of these species, maybe because they are cheap. So now we need to develop marker-assisted selection, so we can increase our works in genetic improvement, and we can foster the improvement of this study like other kinds of fish. So before we use morphology marker, but now we use DNA marker, so we can foster the genetic improvement in aquaculture fishes. For examples, the DNA marker now, this is random amplified polymorphic DNA and restricted fragment length polymorph, amplified fragment length polymorphs, and simple space repeats. So we can go to details of one by one. Random amplified polymorphic DNA, we use only short primers, so we can amplify random DNA segments, anonymous data segments. And if it's amplified in some places in segment, we have products, and if not, they have no products. So we can see one pens here, and B sample is have no pens. So the other one, if they have insertion, one become longer segments and one still short segments. So we can see different different place of this pen in the gel. A and B is different place. So we can make differences between two fish using this. And restricted fragment length polymorphism, we use restriction endonuclease enzymes to cutting the genomic DNA. This, for example, this picture, fish one cut in some places and another fish cannot cut. So we can see in gels after salt and blood, we can see one fish have two bands and one fish have no bands. And also for insertion, one is longer, one is shorter. So use this restricted endonuclease, we can see difference of the difference of genomic encode of the fishes. So another one is amplified fragment length polymorphs. We combine these two techniques together. We use restricted endonuclease, just cut the genomic DNA and put the adapters, and we doing also amplification after that with these adapters, one of these adapters, and then after we provide them in gel electrophoresis, we can see different kinds of bands. So we can make a differences between two fishes or any kinds of fish population. And simple sequence repeat in the end is we use markers, we use repeat sequences of markers, and we need to provide these primers, forward and reverse primers first, and then we can compare with another kind of sequence, the same sequence from the same SSR primers. If we get the different kinds of sequence repeat, we can get different kinds of lengths of the pens. We can see here, and we can make differences between fishes only based on differences of simple sequence repeat. And in the details, what is simple sequence repeat? So simple sequence repeat is repetition of simple DNA like SC, and the SC13, ATA8, ACT7. For examples, this AG repeats and have a sequence in each side of the sequence, and then we make a primers. This is our target primers. And we can classify this SSR based on the sequence repeat as pure, compounds, and complex, and inside that we have interrupted sequence. And the function of this SSR outside the gene and inside the gene is many. We can see here chromatin organization and regulation of DNA metabolic process, and so the most important thing is in gene activity. This is SSR within genes. They can influence the genes. We can inactivate the gene or activate the gene. Also can silence the genes. Also in 3UTR can facilitate the transcription. So for example, this gene is promoter. Promoter is this 1000 base pair outside the genes. This SSR is not inside the genes, outside the genes. This is prolactin, promoter of prolactin 1 gene in tilapia. Is very popular. This prolactin 1 increased plasma osmolarity by reducing kill NAK ATPase activity. And for example, this short SSR in the low expression pair L1, they will increase kill NAK plus activity. And then this better osmoregulatory regulation in cell water and better growth. But for the longer one, CL31, you can see high expression of the L1 decrease kill NAK ATPase activity. And the influence is it can decrease osmoregulation in cell water and reduce growth. So our objective of study here is isolating new SSR primers of rabbitfish and milkfish, and then searching polymorph. And we can use this polymorphism in genetic diversity analysis, strain and hybrid identification, prevents breeding depression, and parentage analysis. And also we want to select the economically important traits like better lipid distribution, and also we want to find fewer intermuscular support of milkfish. This is some kinds of possible activities in marker assisted selection if we can find some polymorphic DNA of SSR in the support of this fish. So we have here experimental procedures around 12 steps to get this SSR. The first is we need to extract the DNA, and then we cut the DNA with enzymes, and then after that we need to ligate with the chain between the segment of DNA with adapter, and then amplify with first amplification of this, and then after amplify we need to hybridize with SSR drops and pull down with magnetic pit spiration techniques. This, and then we amplify again in the second PCR, and we transforms in E. coli, and we doing the third PCR. We want to check if they have a recessive gene or not, and then a positive clones, we sequence it, and we check this the motifs of this SSR inside the inserted DNA fragments, and then we make primers. This some kinds of list of the criteria of the primers. We using online programs, these 3 online programs, and this standards is 18 until 27 base pair, and product size 100 until 450 base pair, and the GC contents is 40 until 60%, and handling temperature is 55 until 60, and we choose low delta G to prevent primer dimmer, homodimer, heterodimer, or hairpin structure. This hairpin structure, and this is delta G free energy. And then we need to begin with GC and end with GC also. And this picture show you how we make primers, design primers. So first we find adapter primer first, where is adapter primers, and then after that we make trial and error for primer paste based on the criteria, and this we make these primers. So these primers after make, and then we evaluate and PCR, and we see if the primers work or not. So here the results. So based on SSR clonings, so our genomic data is quite good for the first steps, and we can see here that we have positive clones more than 90%, for milkfish 95%, and rabbitfish 99%. We can see here that our positive clones is quite high, and also these identify SSR screens. And these screens, for examples, this is sequence for MF6. These clones, MF6 clones, have TCC5 examples, and most of the SSR is twinonucleotide and twinonucleotides. For milkfish and rabbitfish is twinonucleotides. But the percentage of milkfish is higher than rabbitfish. Rabbitfish only 35% and milkfish only 97%. And for summary of SSR analysis, we analyze mononucleotide, dinucleotides, and trinucleotides. So we can see that 93% is most of milkfish SSR is pure SSR, and then more than 50% of milkfish SSR is trinucleotides,